I'd like to start by thanking the organizers for this opportunity to present our work. I am Debswapna Bhattacharya from Auburn University, and today I will present QDeep, our new protein model quality estimation method using deep learning. This is a continuation of our work that was presented in ISMB 2018. Protein quality estimation is a subproblem of structure prediction, in which we are interested in estimating the accuracy of computational predicted protein model. That is, given the three-dimensional structure of a computationally predicted protein model, we like to know how accurate it is without the knowledge of the experimental structure. In other words, we are interested in estimating the quality of the model. This has important implications in structure prediction, including the ability to distinguish between the folded structure and misfolded alternatives, or to select the best model given a pool, and ultimately to help improve structure modeling accuracy. Machine learning plays a very important role in quality estimation methods because it helps to learn the unknown function that maps from protein feature space to a quality score. Supervised function approximation is routinely used for this purpose, that helps learn this unknown function from observed known structures. Let us now take a deeper look at each of these three facets of quality estimation: feature space, function approximator, and quality score, to understand what existing approaches do and how they can be further improved. First, feature space. Existing methods use a variety of different features, including sequence profile-based features, structural features, or statistical potentials. However, they do not use distance-based features. Now, distance maps has recently revolutionized structure modeling. Thanks to deep learning and covariational analysis, we now have the ability to reasonably accurately estimate distance map from sequence. This information could further improve the quality estimation performance. In terms of the function approximator, existing methods use a wide variety of approaches ranging from traditional machine learning algorithms such as support vector machines or random forest to more advanced deep learning approaches such as multi-layer perceptron or 3D convolutional neural networks. But quality estimation could further benefit from advanced deep architectures. One such architecture is residual learning. In residual learning, we predict y minus x from x instead of predicting y from x. Here, y is the output and x is the input. And y minus x is therefore called residual. The idea of residual learning is to predict y minus x and then add x to the predicted y minus x to estimate y. This is equivalent to adding a shortcut connection between x and y. As shown in the figure, this enables the training of very deep network by stacking many layers together. Deep ResNets can be used to further improve quality estimation performance. Finally, quality score. Global Distance Test, or GDT, is a widely used structural superposition-based metric which can be used for estimating how accurate a protein model is by comparing it with the experimental structure, which is defined here. In the equation, the numerator has quantities n1 through n4. They are the number of aligned residues below 1, 2, 4, and 8 Armstrong thresholds. And the denominator has L, which is the length of the protein. GDT score lies between 0 and 1, with higher score indicating better accuracy. Now, just by looking at the equation, it becomes clear that this n1 through n4 are threshold-based quantities, and we could use binary classifiers to model them. And furthermore, since there are 4 of them, we could use ensemble learning for multiple binary classification prediction. This ensemble learning can further improve quality estimation performance. Based on these considerations, our contributions in this work are threefold. First, we introduce distance-based features by distance histogram alignment. Second, we employ stacked deep ResNets for quality estimation task. And third, we use ensemble of 4 deep ResNets at 1, 2, 4, and 8 Armstrong thresholds. The result is QDeep, our new protein model quality estimation methods. Here we show the flow chart of QDeep. It has 4 major parts. In the part A, from a given sequence, we generate the multiple sequence alignment. In the part B, we extract features from the sequence and the model pool. The features include sequence and structure-based features and Rosetta centroid energy terms, which are more standard in quality estimation methods. It also includes distance-based weighted histogram alignment, in which we predict the distance map from the sequence and extract the distance information from the structure and finally align them and use the alignment as a similarity score to be fed as a feature. We use a sliding window of 21 to feed these features into stacked deep ResNet architecture, which is shown in part C. Because we have 4 different thresholds, we use an ensemble of 4 different stacked deep ResNets. Each of these ResNets use 13 different residual blocks with identical architectures. Each of these residual blocks use a combination of convolutional layer, batch normalization, and ReLU activation function. Afterwards, the stacked deep residual neural network uses a sigmoidal activation function in order to predict the likelihood of a given residue of the model to be below the threshold. Acting together, the ensemble of 4 deep ResNets can be used to estimate the quality score analogous to what GDTTS does. Therefore, QDeep score is a probabilistic equivalent of GDT score. We trained the residual neural nets using proteins from CASP9 and CASP10 datasets and validated using CASP11 sets. Overall, there are over 2,000 redundancy removed models with more than 300,000 residues for over 200 CASP targets used for training. Let us take a look at some of the results. In order to evaluate the performance of QDeep, we collect CASP12 and CASP13 stage 2 datasets and compare the performance of QDeep with state-of-the-art methods from CASP. We looked at many different performance criteria, including correlation and loss. Correlation measures the ability to reproduce the similarity score. Here we use GDT as the ground truth and looked at average per target Pearson, Spearman, and Kendall-Stowe correlation, as well as their global counterparts. We also looked at average per target loss, which defines the ability to select the best model from the pool. The results demonstrate that QDeep consistently performs much better than other state-of-the-art approaches. For example, in CASP12, the Pearson correlation, average Pearson correlation of QDeep is 0.74, which is much better than the second best method, ProQ3D, which has a Pearson correlation of 0.68. One of the more interesting things to look at here is most of the top performing methods use deep learning in both CASP12 and CASP13. That raises a natural question as to, are there other deep architectures that are more suitable for quality estimation other than residual nets? To answer this question, we independently train LSTM and CNN architectures using the same set of features and same training data sets as done in QDeep and compare them head-to-head with ResNet. The results are shown here in the CASP12 and CASP13 dataset again. Across all performance criteria, ResNet performs the best in terms of average correlation and loss, demonstrating that ResNet is a very good choice for the problem at hand. One of the other things that quality estimation methods require is its ability to distinguish between good and bad models. We could use a cutoff of GDT score of 0.4 to distinguish between good and bad models. And then we looked at the ability of QDeep as well as other state-of-the-art quality estimation methods to distinguish between good and bad models. We studied the ROC curve and compute the area under the ROC curve for quantitating the performance. Once again, we see QDeep performs better than other methods in both CASP12 and CASP13, demonstrating that it has better ability to distinguish between good and bad models. Finally, we wanted to know whether additional information could help propel the performance further. It has been recently shown that integrating metagenome sequence databases and resulting in deeper multiple sequence alignment could improve secondary structure prediction and contact prediction. Inspired by this, we integrate deep multiple sequence alignment into QDeep and compare it head-to-head with the original QDeep method. The results are shown here, and we could see that deep MSA could indeed drive the performance forward, which outperforms QDeep method. On the other hand, once we extract the distance-based features from QDeep, the performance significantly drops. This demonstrates that distance information is critical to the success of QDeep. But also, additional information such as deep multiple sequence alignment could further improve the performance. With this, I'd like to conclude the talk with some observations. QDeep is really a combination of distance features, deep ResNet, and ensemble learning, which are all three novel contributions in quality estimation methods. QDeep, the results demonstrate that QDeep outperforms state-of-the-art quality estimation methods, and head-to-head controlled experiment reveal that deep ResNet is a good choice for quality estimation performance, at least better than LSTM and CNN. Additional information such as deep multiple sequence alignment could further help improve the performance of quality estimation. In the future, we could extend our method further by modeling high resolution version of GDT score, such as GDT-HA. Finally, I'd like to acknowledge my students, Hussein and Shutanu, who did the work, particularly Hussein, who did majority of the work. National Science Foundation for the funding source, and Alabama Supercomputing Authority for the resources. Our method is freely available at GitHub, including the source code. Please check it out. Thank you very much for your attention.